So in the next slide, in the next part of my talk, I will then address a different point, which is the sex specificity of the pheromone response. We've talked about the control of reproduction and reproductive behavior in the males, but what's happening in the females, and how do animals know what to do according to whether they are males or females. And so what we've seen in the previous part of this talk is that the sense of smell, or at least a portion of the sense of smell, detects chemicals that are essential for the control of mating behavior and provide direct information to neurons expressing the key modulator of reproduction and fertility, which is LH-RH. And the vomeronasal system, as demonstrated by the phenotype of the TRPC2 mutant, provide gender identification, and therefore these two systems are working hand by hand. But this, in fact, is what's happening in the male mouse. And Tali Kimchi, a postdoctoral fellow in my lab, asked the question about female behavior, and in particular, how could male and female trigger different behavior when detecting a similar pheromone, such that a male detecting another male will attack that male, but a female detecting a male will trigger mating behavior instead. So Tali in the lab just performed a very simple behavior experiment, which is she observed the receptivity of the mutant female. So the female mutant is here in gray, and the male mouse is here in black. And what she wants to observe is how receptive is the mutant female to the male approaching her. And as you can see in the video, the female is really behaving very strangely. In fact, not behaving at all like a female, but rather displaying typical sexual behavior of a male. So that was absolutely astonishing, and I actually went and watched the animal and made sure it was indeed a female. It is indeed a female. And when we carefully looked at the behavior of the mutant female, so what you have here in pink is the number of animals presenting mounting behavior towards other females and towards males. In pink are the mutant female, red are the wild type female, blue are the wild type male, light blue mutant male, TRPC2 mutant male. And what is very striking is that the mutant female behaves exactly like the male towards a female intruder, and also is also trying to mate as a male with male intruders. And these are more characteristic of male sexual behavior, that in which you can see again that the female mutant display male typical sexual behavior both towards females and towards male intruders. So that was very, very surprising. Why is the female mutant behaving like a male? We've seen that the male mutant is unable to discriminate the sex of conspecific, so will mate both towards males, with males and female conspecific. But here it seems that the female is not confused about the gender of the other animals, but confused about her own sex identity. And so that was very, very surprising. And we looked at many signs of male typical sexual behavior, and we found that the female mutant display all characteristics of male sexual behavior, the female mutant. We then looked at other type of behavior that are grouped under the term courtship behavior, in particular what we call solicitation behavior, which is the male, normal male, put its snout under the body of the female and raise the body of the female. So this, again, is a behavior that is also highly displayed by the mutant female and never displayed by the wild-type female. And again, it's displayed both towards female intruders and also towards male intruders. So the female mutant both is confused about her own gender identity and behave like a male, but also exactly like the TRPC2 mutant, also seem to have exactly the same behavior towards both males and females. So he's also unable to discriminate the gender identity of the other animals. Another behavior, courtship behavior that we looked at is the emission of ultrasound vocalization. So male mice, normal male mice, emit ultrasonic whistle when they detect a female. And again, we found that the mutant female emit ultrasound exactly like the males and emit them both towards males and female intruders. Now, what is interesting about ultrasound vocalization is that you can look not only at them quantitatively but also qualitatively, which is that if you, this is the sonogram, so the ultrasound are these high frequency pitch emitted by males in response to females, and you can see these complex sonograms here that show pitch jump and very complex sound, ultrasounds that the wild-type female never emits, so there's almost no ultrasound, and whatever ultrasound the female emits are kind of very simple in nature. And what we found is that the mutant female not only emit a lot of ultrasonic vocalization, but also those are quite complex, like the males. So again, for all the type of behaviors that we observed, whether those are purely sexual behavior or part of what I call courtship behavior, the female really seem to behave like a male. And again, you know, this is very surprising. So what's going on? Well, there are two possible explanations of this behavior. One is we are observing a mutant animal in which the vomeronasal organ has never been functional. And because it has never been functional, and because there's a lot of experiments done in many sensory systems that indicate that the function of, for example, the visual system or the auditory system is very important early in development, it's possible that an animal that has never experienced any VNO function has a lot of miswiring in the brain, and therefore the female behaves as a male because of this miswiring. Alternatively, the brain of this animal, this mutant animal, might be perfectly fine, but the phenotype of the mutant just indicates that the vomeronasal organ is important to constantly repress male behavior by females. And so, in order to address this question, we performed a very simple experiment, which is to remove surgically the vomeronasal organ from an adult female. So you can just perform the surgery through the palate, grab the vomeronasal organ, remove it, wait for a few weeks till the nasal cavity heals and the nasal cavity becomes cleared, and we perform behavioral experiments on those animals, and after the behavioral experiment sacrifice the animal and confirm that there were no vomeronasal organ anymore, but that the nasal cavity was perfectly cleared. And by looking at the brain of this animal, we can also confirm that there are no axonal projection to the accessory olfactory bulb. So there's really no VNO neurons anymore. So in this animal in which the VNO has been removed, this VNO-X, what's happening? Well, this is shown in this boxed part of the histogram. As you can see here, for number of animal mounting or the number of the percentage of sexual behavior, these animals without a functional vomeronasal organ behave exactly like the males, and exactly like the mutant female. In other words, the behavior of the male, specific behavior of the mutant female does not originate from a miswired brain, but instead come from the absence of the repression of a male behavior by the female. Now, when Tali Kimchi, the postdoc in the laboratory who was doing these experiments, she also realized something very important to understand a little bit the history of the field. So when Tali performed the surgical ablation of the vomeronasal organ, she realized that when she was performing the experiment, as was described in the literature in all these classical surgical ablation of the VNO, she was not obtaining good results. And she observed that the behavior of these animals was kind of seemed to be suboptimal. And in fact, when she sectioned and observed the nasal cavity of this animal, what she found was that the nasal cavity was entirely obstructed by blood clots. And therefore, it was very likely that both their vomeronasal organ was gone, but also that their olfactory function was impaired because the nasal cavity was just entirely obstructed. And so she modified the protocol, let the animal recover slightly longer, and also aspirate these blood clots on a daily basis in order for the animal to have a very clear nasal cavity and then no vomeronasal function. So it is quite possible at this point that the discrepancy that we have obtained between the result obtained with the discrepancy between the result obtained by surgical ablation that showed defect in mating behavior and aggressive behavior and our results with the TRPC2 mutant might be due to the confusion of really the real sensory defect on this animal with classical surgical ablation, in which those animals were both, might have been both partially at least olfactory impaired and therefore showed abnormal mating behavior together with the lack of aggressive behavior. Now, what is very interesting is that the female without a vomeronasal organ, where the vomeronasal organ was removed in the adult females, also start to emit ultrasound vocalization exactly like the mutant female. And this is quite remarkable because people had attributed the lack of ultrasound emitted by the females by possibly the lack of vocal organs to emit these ultrasounds. So I think it's quite remarkable that after a few weeks after VNO removal, these females are perfectly able to emit complex ultrasonic vocalization. So we've shown, therefore, that these mutant females display male-like traits. And at this point, we were still worried about one particular caveat, which is that, as I mentioned earlier, any type of social behavior, reproductive behavior, aggressive behavior, is really highly influenced by the environment of the animal. So we also observed the behavior of this mutant female in large arena, in which the animal are kept in these 1.2 by 1.2 meter arenas for several weeks at a time in a very enriched environment with little shelters and complex environment, and video recorded for a long period of time. And then we use a software to really analyze in very much detail every single behavioral attempts by the animal. And so this video shows again the behavior of the female mutant in the presence of males. And this is a clip that has been done when the male have just been introduced to the cage. And what you can see is that exactly as was found in the small cages, the females are literally mobbing the male and attempting to show this male-like display with the wild-type animals. So, in other words, what we have observed in small cages seem to hold true also in these large arenas. But what's happening is that when we keep this animal in the arena for a long time, so the wild-type male and the mutant female, the male becomes more and more aggressive and ultimately mates successfully with the females. And so all the mutant female end up being pregnant. And so that's actually then enabled us to perform another set of observation, which is the maternal behavior of the mutant female compared to the heterozygous as female. And what we found was quite interesting, which is when we looked at the amount of time the females spend in the nest. Mice have communal nests in which all the pups are put, and then the female just take turn around the nest. We found that, as expected, the non-mutant females spend a lot of time in the nest, but the mutant female is spending less and less time, and in fact, at some point, even we removed the divider between the mutant colony and the non-mutant colony, and the mutant female immediately abandoned the nest to explore the new territory. So these mutant female therefore have a very clear defect in maternal behavior. So that's very interesting. To summarize, the mutant females show male-like behavior and show a defect in female-specific behavior. Now, all these type of behaviors are known to be under the influence of steroid hormones, and we therefore wonder whether the level of steroids were different in the mutant rather than wild-type animal. And so when we checked the level of testosterone or the level of estrogen in the animal, we actually found that there was no difference between wild-type animal and mutant, which is that both for males and females, female mutant had female-specific levels of estrogen and didn't have elevated level of testosterone, and therefore one could not attribute the male typical behavior of the mutant female to abnormal level of testosterone. So, but again, this is quite surprising because it entirely challenges the ideas that we have now regarding the male and the female brain. So the assumption regarding the difference in behavior between males and females are as follows. So it is thought that steroid hormones have two roles in establishing the male brain. One is that steroids are elevated, and testosterone in particular, around birth, and this has an organizational effect such that specific set of connection and nuclei can be formed in the developing male brain, and that in the female, the absence of this surge in testosterone leads to a different type of differentiation, which is typical of females. And at puberty, a second surge in testosterone leads to the activation of this male-specific circuit, whereas in the female steroid lead to deactivation of female-specific circuitry. But what we found is that, in fact, the male adult female without a functional vomeronasal organ, whether this is come from a genetic mutation of the surgical ablation, is perfectly able to display male-specific behavior. And therefore, we have to draw this circuitry quite differently and assume that in the female brain are both functional, potentially functional male circuit and female circuit. And those are under the control of a switch, if you wish, that is controlled by the vomeronasal organ. And therefore, the vomeronasal organ enables the female to have a female-specific behavior and represses a male-specific circuitry that does exist and is potentially functional in the female brain. And if one removes the switch, the vomeronasal organ, either genetically or surgically, then one can observe this potentially functional male circuit. Now, there's a lot of attraction to this model. One of them is that although people had postulated that male and the female brain had very different circuits and nuclei that would explain the difference in behavior between males and females. Well, it just happened that people had serious trouble to identify circuits and nuclei that were distinct between males and females. People have looked for decades to the anatomical correlates in the brain of male and female specific behaviors, and all what was found was slight differences either on the size of specific nuclei or the composition in neurotransmitters and other type of small quantitative differences, when the observation of male and female specific behavior shows that differences must be qualitative differences and not only small quantitative differences. So in contrast, a model like this, in which both males and female circuits exist in the brain and are under the control of a switch, a sensory switch that is itself sexually dimorphic, that is a very attractive model. Now, obviously, for sake of balance, one has to postulate that in the male brain there is also both a male and a female circuit that are both potentially functional and are under the control of the vomeronasal system. So is this reasonable or not? Well, very interestingly, some old literature shows that if one remove the vomeronasal organ from an adult male or impair specific nuclei of the vomeronasal system, then male rats appear to suddenly display maternal-like behavior, retrieve the pups, bring the pups to the nest, build a nest, and have a pseudo-nursing behavior that is never observed in the wild-type rats. So therefore, the removal of the vomeronasal organ seem indeed to let female-specific behavior emerge in the male, and therefore, for all intents and purposes, I'm going to suppose and I'm going to postulate that the male, like the female brain, have both male and female circuits that are potentially functional and themselves under the control of vomeronasal system, vomeronasal function. Now, this is a very attractive model, also makes a lot of sense from a developmental standpoint, because an entire circuit that is built differently in the male and the female is quite difficult to conceive. It's difficult to build an entirely different set of connection in the female brain and in the male brain. This, in contrast, seem to be much easier. There is one brain that is built under a very complex set of genetic information, and then there is a switch, the repression of the wrong behavior and the activation of the right behavior that is performed by one specific system, which is the vomeronasal cues. Now, this model has quite a number of implications from an evolutionary standpoint, and I would like to finish this presentation by what I think are a number of really interesting speculation of what the role of a system like this could be during evolution. One of them is this system really provides quite a lot of flexibility to evolve certain behavioral traits in animals that are very close genetically. And all you need is to have an animal that, for example, in female will take one part of the circuit that is usually expressed by the males and then suddenly activate that circuit, and vice versa for the males. So in other words, you would have some type of inversion of some of these arrows here, such that, for example, a male could, in a species very close to another one, suddenly express a trait that is seen more as a female trait. And this does exist in nature and has been described, in particular in this very interesting animal called the vole. There are two types of vole, two species of vole that are quite close genetically that have been described, the mountain vole and the prairie vole. Now, the mountain vole, the males are promiscuous, they mate with a female and then abandon the nest. The prayer vole, in contrast, the male of the prayer vole has a very different behavior. The couple are totally monogamous, and the dad is taking care of the pups and have a lot of parental traits that are seen in the female in the other species. And so one can see that this could be some type of part of the maternal-like circuit that has actually been adopted as a male-like behavior in the prayer vole compared to the mountain vole. And you know, we can imagine like these animals that are very similar to each other genetically could evolve specific traits that are thought to be classically displayed by the other sex. We know, and this type of behavior or evolution would be extremely hard to explain with the classical model of male and female brain being so distinct. We know some of these examples, the old dads and the new dads. These are obviously a new generation of humans' behavior that behave a new generation of humans that behave very differently than their dad or granddad, which assume that taking care of their children would be absolutely non-male-like, and this is obviously complete speculation and more presented here as a joke, but it's very clear that one can, in the course of even 1 or 2 generations, have a very different type of behavior, and these obviously could very well come from a lot of flexibility that is offered by these models. Now, what we've seen are very small changes in behavior, so somewhere in part of this circuitry. Something more profound could happen, and this would be an entire change of the entire repertoire of behavior. In other words, a female now behaves exactly like a male, or male exactly like a female. And it's interesting to look throughout evolution that there are indeed animal species that do that constantly. So there are specific species of fish that in which the female switch into male and then from male come back to a female behavior. And again, this would be extremely difficult to explain if one considers that the male and the female brain have very distinct behavioral circuit. It's much easier to explain if one considers that the brain in each species is essentially bisexual and there's just a switch that needs to be modified. So I mentioned that again in the fish. There are a certain number of fish that can switch their behavior from males to female, sometimes in a matter of days, sometimes in a matter of hours, sometimes even in a matter of minutes. And so again, I think these schemes provide a very interesting template to explain this type of behaviors. A little bit further in evolution, a very fascinating model is the model of the whip-tailed lizard, in which this particular species of lizard has only females that reproduce by cloning, so in a sort of pathogenetic manner. And what is interesting is that the females within this species adopt a female and then a male-like behavior, although there are only females within the species. And these depend on the specific level, the ratio of progesterone and estrogen. And one of the female will adopt a male-like behavior that will help the other female to ovulate, and then in turn the animal that has a male-like behavior now will adopt a female behavior, etc. Now what is quite interesting is that there is still the ancestral species in which male and female still existed, and this is the case here. You have a closely related species from which the parthenogenetic one evolved, and in here you have a male and a female, that show a specific male-female sexual behavior, and the two females of the parthenogenetic species do behave and show exactly the same traits, showing that these females have kept both the male and female behavior secret and are just able to switch from one behavior to the other. And this work, fascinating work, has been done by David Crew in Austin, Texas, and his group. And this is an absolutely fascinating model. So just to conclude this talk and to talk about our future plan, I think that this new model of the male and the female brain that extend from the fish or the reptiles to mammals is now opening really new avenues of research on the control of male and female specific behavior. What we are interested in doing in rodents is to look at this model in a more mechanistic way, which is what specifically are the switches, what are the brain areas that function as switch, how do they function, this sexually dimorphic area here that control the downstream male and female circuitry, what are those circuit components, how do they operate, and can we have a switch that operates in a matter of months, weeks, days, hours, minutes? These are all mechanistic questions that we would like to address. Thank you very much.